Should I start? Yeah. People can still come and join us because it will take a few minutes. I would like to welcome everyone to our exciting webinar hosted by the Cytomics Center of University of Exeter. And I'm really excited and delighted and really happy that Mike Stadinsky, our speaker today, the COO from Phytonex, agreed to give this fantastic talk about new technology and about new innovations in fluorochrome design. But before he's starting his speech and talks about this new technology, I would like to give a short overview about Mike's biography, because me and Mike, I think we know each other for a while now. We had a nice collaborations and co-marketing together. At the moment, he is the COO of Cytomics. He was the former YP GM from informatics section from Becton Dickinson and Company Life Sciences. He was also the former COO of Flojo. That was the time when I met Mike the first time, and I can witness he really led Flojo and transformed Flojo into the leading company of single cell omics, I would say, because Mike likes to play with numbers, not only on flow cytometry side, but also on the genomic side. So he immediately really brought Illumina collaborations and brought Illumina on board to set up the gene expression platform on Flojo. So I think that was another exciting moment for Flojo. And now I'm very glad that Mike agreed for this session about this new technology, especially at the moment. We have also the spectral flow cytometer with 40+ colors, and I'm really excited to learn more about the new type of dyes, how we can expand our panels on a single cell level. Mike, it's your turn. Glad to have you here.
I really appreciate the introduction and the invitation to speak, Rafe. So it's great. And for the audience, we do like to keep this interactive. And so I'm joined by Sid and Thomas, who's a research application scientist with Phytonex, and she led a lot of the 40 color work. She led all of the 40 color work that we'll talk about at the end. And so again, feel free to use either the chat box or the Q&A function while we're going. And she'll interrupt me and we'll have discussions. And it's honestly a lot more fun that way. And so just a bit about Sid. She earned her PhD in medical sciences, immunology, and virology from Harvard. She's got over 20 years in flow and immunology. And so she's published over 34 papers, received an AI Young Investigator Award, as well as the President's Rising Star in mucosal immunology. So she's an awesome partner in all of this. And so I'm really excited to show you this. And we have a lot of fun on some of these webinars, particularly answering some of the questions live. And so as you think of things, please put it in the chat box or the Q&A. It makes it honestly a lot more fun for me than talking essentially to my own reflection. Although I get to see Rafe. So I'll go ahead and share my screen here. All right. Great. Hopefully everyone can see my screen. So let's go ahead and get started. So, we've been looking at a lot of exponential graphs really these past 2 or 3 months. And here's one I think we're probably a little bit more familiar with, comfortable with, which is looking at the dyes that have been achieved over time. And I'm going to add some perspective to this. And I think it's really keying off of really the introduction here, which was, let's talk about new dye technology. Let's talk about where we're really held up in some of the infrastructure and trying to push it. And then we will obviously spend a good amount of time too talking around spectral flow cytometry, but we'll also talk about how dye technology is really holding us back across a whole set of instruments, right? And so what we're looking at here is really the number of colors doubles every 10 years. I'd say we're right, and we'll talk a little bit about this as well, we're right on the cusp of essentially breaking that in terms of putting out more dyes at one time than have been put out in the past 10 to 15 years. But I'm going to explain why and how we get there in a moment. Let me just make sure that everyone can see. Okay, cool. So anyway, when we look at this, actually, there's another way of looking at this, which is what if we looked at this through the lens of what the relationship with hardware development was? And so that's how we'll look at this first. And so really, when we think about it, dyes are really driving hardware development. When we get the development of the first tandem dyes, we get the development of a 12-color Fax Vantage. When we have the quantum dots come out, then we get the development of an 18-color LSR2. And if you think about it, this occurred again because we had the brilliant dyes come out, the brilliant polymer dyes, and then we had the development of the facsimile, really, if you will, catching up to that innovation. You know, the other part of it is, if you think about it, as we move up and up and up into the detectors, then you get democratization, right? And you get benchtop instruments like the Cytoflex or the Attune, who can come in here at a 12 or 18 color level and perform really well at, if you will, a lower number of parameters. Now we're in an inversion, and in fact, we just spoke to this when we're talking around spectral flow cytometry, which is that we're over-detectored now, right? We have far more detectors than we have dyes. And part of this is because of spectral, but the way we see the universe really is, we have these, for instance, a Cytec Aurora that's got 5 lasers, 64 detectors. We'll show you some of the data that we've put out there and also shared, both the raw data and the analysis of, where we're working our way up into 40 colors, and we'll describe how we did that, how we thought about that, and what that effort looked like. We also have this ID 7000 from Sony that's got 7 lasers and 188 detectors. Now you have this Bigfoot sorter that's got essentially 9 lasers, but really more functionally like 7, right? And yet, we see this fundamental limitation. We're held back in the maximum number of colors, right? And yes, there's spectral instruments, but we'll speak to this. We see this more as, well, these are detectors to be hit very cleanly with fluorophores. And again, something that we'll touch on quite a bit. And so we'll talk about flow cytometry panel design with NovaFluors. I'm gonna try and step it across multiple different sets of instruments. And then we'll talk to you around some of the applications that we're taking a look at. And again, spend some time too on our 40 color effort. And so to take one step back, how do we think about panel design, and where did we focus the efforts that we've got in new dye design first? And I think the first thing, and I think even above all of the different considerations that we put in a panel design, is really paradigmatically what we think about is we try and ask a biological question, and then we have to deal with all the compromises of the underlying infrastructure of fluorescence to be able to answer them. So it's a bit of an inverted relationship, if you will, even between fluorescence and biology. Really, ideally, we would have fluorophores that were designed for biology, and we'll talk about what that means. But start thinking now, what if you had that, and what would your requirements be? What if you could design one from scratch, which is a question that we'll answer actually in just a moment. But in terms of panel design, right, we have to deal with spectral overlap. I always say, our current fluorophores lead a decidedly more colorful life. We have to deal with spillover spreading, right? I honestly think that this is a very incredibly pernicious issue, right? This is something that affects every single flow cytometry experiment. Running a spectral flow cytometer really doesn't get you away from this. If anything, you actually get, you get quite a bit more spread contribution because you can use more and more and more dyes, and their performance can be quite bad. So it's something that we'll mention, and we'll actually use that as a way of saying, what can you do in terms of adding new colors? Staining index, or we'll actually talk about this in the context of also something called separation index, which we think is a more objective way of saying, can I separate my biological populations, which is really a better objective notion of brightness. And then finally, one that I think is the closest to the biology is antigen density, right? And if you think about it too, even with antigen density, we take a fixed brightness fluorophore and we map it into a, for instance, something like PE or PE Cy5, and we map that into a low antigen density marker, right? We've never said, well, I have got something that's at this antigen density. I really want something to have a specified brightness. We can't do that across multiple different colors. So we're still stuck, and dye performance affects every single part of this. In fact, panel design is dealing with essentially and compromising with dye performance. And so where have we focused our efforts, right? And so we can sort of set aside for a moment the deep UV. We can set aside for a moment the IR laser, right? There's very few that are out there. And again, we focus in on, of course, some of these first 5 lasers, the first five lasers that are in fact, on a Symphony or an Aurora instrument. And in fact, we focus even further down into this blue and the yellow green lasers, because it's an area that just hasn't seen innovation in several decades. And why did we focus there, right? So if we look simplistically and say, look, let's look at dyes that have a primary excitation laser off the blue or the yellow green. First of all, you can see that they're rather old with the exception of brilliant blue, but the amount of cross excitation that occurs, something that we'll show you and quantify again in a moment. You got per-CP that's cross-excited off the violet and the yellow green. You got the brilliant blue dyes, which, as we'll show, have large amounts of cross-excitation off of the violet. You got PE and the whole PE family of dyes, right, which certainly has excited primary lasers off the yellow green, but as we well know, has a cross excitation off the blue, right? And this is something where we focused quite a bit of our effort on, right? And we think about even instruments that have been designed, something like a, Fortessa, right? They have a trigon off the blue. And that's specifically because, well, we've been dealing with PE and PE conjugates. There wasn't anything really else to detect there. We're going to talk about how we've really focused in on these laser lines because they were in desperate need of innovation. There's another point to make, because this is a little bit more than just cross excitation. And it certainly affects, things when we start looking at dyes in a spectral environment. And so here I'm showing a ribbon plot, and this is just looking at the fluorescence intensity across the entire spectrum of, this is just a prototypical dye at this point. And the reason I'm showing this is because you certainly have a primary detector. And in conventional cytometry, we would say, well, that's the detector for that dye, right? Of course, spectral cytometry, attempts to look at the signatures and then deconvolute these. But I think what's really important to capture is that all of the other places where you're seeing fluorescence across the spectrum are introducing spread. So while you can deconvolute these dyes, you are making the analysis, if you will, much more difficult. And so again, this is something that I'll show you in the context of our labels, how we think about spread, and then how we think about upgrading panels, but keeping spread essentially constant or keeping it very, very low. And so I think this is important. It's also because this is something that we focused and designed for, right? And so let's talk about current dyes, and let's talk about the way we like to think about it. So again, showing these ribbon plots off of a 5-laser Cytec Aurora, we're looking at these dyes, if you will, their signatures across an entire spectrum. And so we can see per CP, has this primary detection again in the blue, but honestly has a very, colorful life, right? It's cross-excited off the yellow-green. We can see some cross-excitation off the violet, right? It's contributing spread. Brilliant blue, obviously a very bright dye, but as we'll discuss, is very noisy in the violet. And then similarly, Brilliant Blue 700, I mean, you can see that it certainly has a primary detection in the blue, but is arguably a red and also violet dye, right? And so when we think around designing a fluor for, we consider this quite a bit, right? Which is how much cross-excitation can we engineer out? Can we contribute 0 to no spread everywhere else, right? Because really in an ideal world, you would have just the primary detection and then no contribution everywhere else, right? That would be a theoretically amazing dye. So let's actually take that up. And so this is the question that I think we're really driving towards. When I talk about all this background, it's like, okay, where's this going? The reason I'm talking about spread, and I've talked about these issues with panel design, is to get in our mind, what if we could start from scratch? What if we could take all of the things we thought about with dyes and set them aside and say, okay, what if you could just design a fluorescent label? What would it look like? And then given that, what would panel design be? So we're sort of picking up that thread of, okay, well, the first one would be pretty obvious. We were just talking about it. It would be bright and clean, right? Ideally, it would hit its detector incredibly cleanly and contribute no spread anywhere else. It would also be silent in the violet. And this is something that, as we started talking to more and more customers, became very important. We want to use the violet. We want to use that for more phenotyping, right? And so we really would like these to be silent in the violet and contribute 0 spread into the violet, right? Not be cross-excited. And the other part of this is limiting cross-laser excitation. Think about where we're trying to target in the blue, yellow, green. If we could limit, for instance, we could have a set of yellow-green dyes that are not cross-excited off of the blue. Well, then we could leverage all of that yellow and green and blue bandwidth, if you will, that we have across all of our cytometers. And that is across every cytometer. The second part of this is really emergent. This would be the second requirement that you would have, right? Which is the practical part of this. Are they consistent? Are they stable? Right? And I'll share with you, we've been ordering pretty large amounts of antibodies, and we're dealing with the same kind of issues of, when we order from other manufacturers, like whether it be lot-to-lot consistency or stability, or practical items like I need to stain 15 or 20 minutes before, right? Or I need to use special buffers. We're going to spend a good amount of time on that because I think it's practical reality of how you use fluorescent labels. And it was honestly a result, some of the data I'll show you is a direct result of some feedback that we had in webinar sessions like this, right? Where people said, this is great, but show me how these things work in the real world. So we'll discuss that. And the final part of this is the new to world capability, which is controllable brightness, which asks the question, and I'll show you, what if you could take any color and actually tune its brightness? And what would that enable for us in terms of antigen density? So we'll touch on all 3 of these, and I'll show you data for each one of them. So how do we do any of this, right? So what we have is actually a platform for designing new fluorescent labels. And the way that we do that is we take these small organic dyes, we bind them on single-stranded oligonucleotides. And these we can actually self-assemble and anneal them into this phyton structure, which is a cruciform of DNA. And this cruciform provides us a scaffold on which we can design new fluorescent labels. And if you think about the way that we can do this, we can actually engineer down to the sub-nanometer level the way that these individual component dyes work with one another to get fret, right? And so fret scales with the square of distance. So it has a huge impact on our actual ability to tune the way that you're getting energy transfer in a fret interaction. But one point here, these are not tandem dyes. We rely on the energy transfer of fret, but they're not tandems, right? One phyton dye is one phyton dye, and it doesn't change when you put on different antibodies, right? And in fact, there's no tandem degradation, because if you think about what we're looking at here, we have a very, and I'll show you, a very stable platform of DNA. The other part of this is that this platform provides us 4 different arms, 4 different, if you will, individual scaffolds, which we can use to tune brightness up and down. And again, I'll show you data on this. There's 2 other parts that I really found interesting when I first started looking and working with this technology, which is, one, you also have the flexibility of having a single strand of DNA come off of the end of this phyton structure, right? And we actually, in the introduction, we were talking around genomics. There's actually lots of interesting applications there that we can go after, but it also enables us a really flexible platform where we can take a single strand of DNA and use that to conjugate it to the antibody. And so again, we'll discuss how we're doing that in a moment. And the other part of this is that we're relying upon oligo manufacturing to make these. And so again, I'll show you what lot-to-lot consistency looks like, but it's incredibly consistent, right? Oligo manufacturing is very well known, very well understood. So let's focus first on this notion of bright and clean, right? And so really what we've done, again, going back to our blue, yellow, green focus, is we've gone in and separated the excitation of the blue and then the yellow, green lasers. And so what we thought about is actually going in there and stacking blue and yellow, green detectors. And these are our first 6 labels, right? We've got a whole large number of new labels actually coming out in actually just under a month. And so, because the other part of this platform is it gives us the rapid ability to vet and create new fluorescent labels faster than really anyone's come out with them before, certainly within Flow. And so what we've done here, you can see we've actually separated the blue excitation, right? And we've got the yellow excitation really off of another line. And so we can go in and use each of these laser lines independently. You can see too that we've stepped the Stoke shift out here for these labels. I know it's sort of hard to take that in webinar context. And so what we've done is partner up with some folks so that you can also use their tools to examine this in your own panels, right? We've also published this up on our website. So these are resources that you can use to take a look at, okay, well, how would I be able to use these to upgrade my panel on a conventional flow cytometer, or as we'll discuss in a spectral context. So we put them up on a resource called FPBase, which is an open source spectral viewer. It's quite nice. It's got all of the fluorescent proteins in addition to a number of different dyes. It's also up in Fluorofinder. Also, the folks at Cytech were kind enough to put our labels up on their full spectrum viewer. And I think this one honestly gives the clearest indication of what we're up to. If you remember back to our discussion of, man, it'd be great if we just had a dye that hit that detector really, really cleanly. You can actually see that's exactly what we're doing here. As we'll discuss, very clean in the violet. And I'll show you what that performance looks like. We can hit these, our first 6 dyes hit these detectors incredibly cleanly. And again, we'll discuss what's going on here in the red. I'll just put a pin in that for now to say APDs are very sensitive in the red. And the gain on those is turned up very, very high to separate 2 different red dyes, but we actually see no spread contribution or cross excitation over there. And so what do we mean by stacking? What could you do when you have labels that emit cleanly? And the answer is, okay, well, let's take a look at it in the context of, I'm going to go design a conventional flow cytometry panel for an Attune or a Fortessa, right? And what would the manufacturer's recommendations be? Well, we can see they're somewhat redundant, and it's because of the amount of cross excitation that we're seeing, right? We see PE that shows up in both of these channels because it's cross excited off the blue. Same thing for PE Dazzle and PE Texas Red. If you use PerCP Sci5, then we cannot use PE Sci5. So you can see that immediately once we actually start trying to do some biology in the context of these traditional dyes, we're immediately dropping our capability and the number of questions we can answer down by at least 2 to 3. And now we're only talking about what, a 6-color panel. We go in there and we can actually stack each of these detectors very cleanly. And this is what we mean by stacking. We're able to put these into these detector lines and use them simultaneously. And so I'll describe some examples on this. Just to drive this point home just a little bit more, what we'll do now is just compare this to PE Dazzle. Right, it's PE Dazzle, right? Bright dye, very well-known dye. If you're using something like PECF 564, it's the same dye, and we'll actually show some performance characteristics versus this across the entire spectrum in a moment. But I think what's really important to consider is, PE dazzle has this very promiscuous excitation of 48, the 532, and the 561 line, right? Whereas what we've designed is a Nova blue dye, right, that can be independently excited off the 488, and we can separate the Nova yellow so it's excited off of the 561 line, right? And so that's what enables us to stack that. So I think, again, getting back to this notion of spread and what's the data analysis going to look like, I think one of the most powerful visualizations of this is actually probably the simplest. And it's probably the simplest slide that I have, but I think it's one of the most powerful. Because what we've done here is take single stain controls of each one of these labels, right? Each one of these was run on CD4, right? And then we concatenated them. And we said, after compensation, what does spread look like, right? And so it's particularly off adjacent channels, right? If you were to run PE dazzle and something in an adjacent channel, you would see this trumpet effect, right? You would see this effect where you get this large spillover spread. And in fact, even off adjacent channels with our labels, right? This is a dye that's excited off the blue and emits at 530. That's the way we name our dyes for what it's worth. This is excitation. And then that's their emission peak, right? You can actually see that even in the adjacent channels, we're not getting really any significant spread. And so, it's another thing that we're driving towards. And again, we'll look at this in the context too of the 40 color data, which is this notion of high resolution biology, where we have this high resolution of biological populations because we're not contributing spread, right? And so, again, a theme that we'll pick up throughout this. So how do these, a natural question, how do these compare to dyes that I'm already using that are out there in the world? And so we got 2 of our dyes to the NIH. This was early on, and they took really through a huge amount of dyes up against our Nova yellow dye. And then we also were looking for an apples to apples comparison for brilliant blue dye versus our blue dye, right? And we ran these on beads and PBMCs and really looked at both spillover spreading, cross excitation, as well as brightness. And so let's dive in on that. And I think it's a great proxy for one conventional cytometry, but it gives us the first indication of what's going on when we start looking across larger detector arrays. So this is not a ribbon plot, so we're looking at this in a bit more of a traditional way, where we're looking at 5 detectors off the blue, 8 off the violet, 7 off the UV, 3 off the red, 5 off the green. And in this case, what we're doing is comparing brilliant blue 630 in this dark navy color versus Nova blue 610, which is in this lighter blue color. So you can see that in the primary detector, right, the Nova blue is not as bright as the brilliant blue. And again, we'll pick this up, the notion up of brightness in a moment. But notably, it's much, much cleaner across the spectrum, right? So there's a cost of doing business, right, if you will, when you're using a brilliant blue dye, which is that it's mostly a violet dye that happens to emit in the blue, right? It has a huge amount of cross excitation in the violet, right? And it's got quite a bit of cross excitation as well off of these green detectors. And so you have to deal with what's the cost of doing business with that. Whereas we can hit those detectors incredibly cleanly. What about the Nova yellow? So in this case, we compared Nova yellow 610, same setup, same instrument, and compared it to Alexa 694, this PE CF594, which is PE dazzle, and then this ECD dye. So you can see that we're brighter than Alexa 694, nearly as bright as this PE dazzle, and brighter than ECD, but notably, again, much, much quieter in the violet, right? And I think this is incredibly important, right? Less cross-excitation, much less spread, right? So incredibly interesting performance profile, because now you have these dyes which can, again, hit that detector very, very cleanly and have low cross-excitation and low spread contribution. Again, we'll quantify that in a moment. What about brightness? This is something we've had tons of conversations about. And for us, I always like to joke that, like, I'm a recovering biologist, Seton's a real biologist, right? And as biologists, we want to just separate a biological population. That's what matters, right? If you told me something has high quantum efficiency, I don't care, because if it has high background, I just don't care, right? I do deeply care that I can separate a population from its negative and separate it from background. But again, let's think about this again. Let's quantify it. If we can increase the brightness of something, but then we increase the background substantially while we do that, then actually our ability to separate the population hasn't increased. And so we were pushed to say, let's look at this through the lens of separation index. And the reason that we're doing that is actually what we're looking at is we want to have a dye that we can separate from background. And in fact, you can see that we're looking at the 84th percentile of the negative population. And we want to have very high separation of that positive population from the background. And so that's the way that we like to think about brightness. And again, this is one of those discussion topics where, you know, feedback and ask questions around this, because I think it's an important topic for us to think about and say, okay, objectively, what are we talking about here? And so what we found is that the separation indices of these dyes that we got to the NIH had the same separation index as APC. And again, this is something to put a pin in, because we'll also bring up later how we were able to look at low antigen density markers like chemokine receptors in the context of our 40 color experiment, right? So it's not just looking at CD4, for instance, all the time. And spread. This is such a, we keep coming back to this because it's like, you know, we run the panel, we're really excited, we run 30 color experiment, right? And then we get the data. And you know, as Rafe knows, and as Seddon knows, and I see some familiar names on the some familiar names on the webinar. This is something of an obsession of mine: is how can we make the fluorescence, make the data analysis easier, and make the data easier to interpret? Because all of our algorithms, all of our gating, all of it comes down to what that data looks like in the end. And I think what's exciting about this is when you look at this Nova yellow dye, you've got the lowest spread contribution compared to 30 other dyes, right? And so you have this dye that can behave incredibly well in terms of its brightness, right? But is well behaved in the sense of it contributing the lowest amount of spread. And the Nova blue dye, honestly, compared to this, in this apples to apples comparison versus Floating Blue 630, is a really incredible performer because it has much less spread contribution. So what does that mean? And so we'll focus first on this conventional cytometry panel, and this is some work that Seddon's doing and actually analyzing the data on now, where we're actually able to upgrade conventional panels, right? So we've actually done this on, in this case, in Attune. We've done a number of these with customers on Symphony instruments, or other instruments, something like Cytoflex in the mid end, right? And so, and what does that really mean? And what it means is looking at a panel and saying, okay, well, what compromises would I have to make just to be able to answer my biological question? So in an ideal world, I'm asking questions around memory T cells, I would be able to have all of these antigens in my panel, right? But I immediately run into a problem. I obviously can't use PE dazzle off the blue and the yellow, and I certainly can't use Percy P-Sci5 and PE Sci5 together. So I'm immediately down 2 questions, right? It's like fighting with one hand tied behind your back, because while you have 13 detectors off of these instruments, you can only ask 11 questions, right? So maybe what you end up doing is running 2 panels or 3 panels, right? And so what we're saying is, well, that's not necessary anymore. And so what you can do is actually go in and stack these, right? And so this is what exactly what we've done. So we've gone in and put our 3 blue dyes in here, our 3 yellow dyes. We drop PE Sci7 down, and you've got a panel where you can maximize the capabilities of a conventional cytometer. And I honestly think this is really exciting. We've worked with a couple of customers that are working in this kind of context, or in the context of a higher detector instrument, and we see their panel and we say, look, let's replace 2 of your labels, and then we can also enable you to ask anywhere from 3 to 4 new questions, right? And so I think that that's an incredible story, because it's, instead of having to run 2 or 3 panels, you can get them all answered in one. And I think that's really exciting when we think about how we can leverage our current capabilities and our current cytometers. And then we'll talk too, of course, on what we can do in terms of spectral. The second part of this, I think, is really interesting, and it's something that we were pushed pretty hard on, which is, all that's great. New colors, they're very clean. Got it, totally understand. How hard can I push this, right? And what are you doing that's different compared to what we're using now? And so I think these are really interesting questions, and they were completely a result of discussions we had, right? Be they in person, back when that was a thing, and webinars. And so we've done some work on this, and actually continuing to do quite a bit of work on just how consistent and stable can we make the platform. So the first part of this is probably the simplest experiment, and it's one that we're running in a bit of a different format now, but that is we just leave our labels out on the bench under tinfoil. So quite possibly the easiest experiment ever. I think Seddon jokingly calls this the absent-minded graduate student experiment, but I think it also addresses some needs that a high throughput community has, which is that you can actually leave our labels out on a bench for out to 3 weeks. That's just the longest that we've tested to date. And there's no change across the entire spectrum. What if we can push that harder, right? And so what we did is we actually took PBMCs with conjugated CD3, conjugated to our Nova yellow dye, and fixed them at 2% paraformaldehyde, and literally left them in the fixative, right? Which I think is a pretty intense, very abusive experiment, right? left them in the fixator for 2 weeks, and then came back and ran that versus freshly stained cells. And there's no difference, right? And so we didn't realize when we ran this really at the end of last year that this was going to be relevant to our current working environment. But you can fix these labels, and there's no performance change over 2 weeks, right? And this is just the longest that we've tested. There's two other points to make around this. Literally, tinfoil is the light protection device here, right? That we're not doing anything special, and there are no special buffers. This is all just in, this is PBS, paraffin, and we've tested parafformaldehyde and as well as paraffin fixation. That's it. There's no special magic going on. And the reason for that is that if I remember I showed you the cartoon of this DNA structure. It has a natural curvature to it, and that stabilizes those component dyes on there, but it also provides essentially a solubility shield, right? So it holds water to it. And so we think that that's actually what protects these fluorescent labels in solution and really adds a lot of stability to them. So that's the stability, and we're actually pushing that now, and we're taking this into new applications, and be great, happy to have a discussion around that. That's, again, some work that's actually going on right now in terms of, okay, now that you know this, where can you take the platform inside, on the cell, et cetera. But the other part of this was, as we started designing larger panels, started dealing with more and more antibodies, was dealing with lot-to-lot consistency and actually taking that head on. There's a couple different points to be made here. The first one is that we share our lot-to-lot data. So our spec is that and this is across the entire spectrum, we design and manufacture so that there's less than 5% variability lot to lot. And in fact, what we see here is a lot less than that, right? And so these are actually two different lots of our dyes, less than 3% variability off one dye, essentially none off the others, essentially overlays. And then this is less than 4%. And I think, this is important because this is the manufacturing view of the universe, right? Less than 5% variability, and we're always going to do that. But there's another part of this, which is that we run each of our lots on flow cytometry, right? And so we actually have a QC on cells commitment, which is that every single time that we produce a new lot of dyes, we put it onto an antibody and we run it on PBMCs, right? And so that's an incredibly important thing for us, is we want to use every one of these in our applications. So we've tested it, have seen CVs under 4%, right, in between lots. And then we've also sent these to an outside laboratory, and there's no statistically significant difference between the staining indices. So we've gone all the way out to literally the full extent of the biology, right? Which is just how consistent can we get? Can we make lot-to-lot consistency in fluorophores and some of these issues that we've seen in other technologies? Can we make that a thing of the past, right? And it certainly affects our experiments, but it has a huge impact, right? When you're thinking around, I want to use this fluor and I just want to use it for years and always count on that data, right? So honestly, pretty exciting stuff when we think about that. And again, we're very open about this. The other part of this was just more dealing with our own frustrations, where every technical data sheet that we make has, that label run on cells. So maybe I'll pause here, and just see if there's any questions, or open up for that, and then we'll dive into controllable brightness and some of the 40 color.
Yeah, no questions thus far, but yeah, please throw them out there. Oh wait, we've got one. Can you use these dyes with viability and DNA staining as they contain DNA?
Yes, no, so sorry, yes, I like this question, sorry. No, you cannot. So the viability dyes that stain DNA will definitely stain this platform. There's no protection, if you will, for, intercalating dyes, et cetera. What we use is the fixable dyes, like the amine reactive dyes, so the non-DNA based dyes, when we're using viability staining in our own panels.
Oh, and one other one. They were wondering if we had tried our dyes under several stimulation conditions with PMA ionomycin per 6 hours or CD28. I'll answer that because Mike doesn't know, but I was running that last night, and I actually have to acquire the data this afternoon just because I ran out of time on the flow cytometer. So I will be able to answer that soon.
Yeah, that's great. Intracellular as you can tell, is a very hot topic for us. And so, maybe it's worth just diving in a little bit on that in terms of the size and what we're looking at. So maybe I'll speak to the size set, and then maybe you can describe a little bit more about what we're looking at.
Sure.
Cool. And so the size of this platform, it's about the same size as the antibody, so it's 150 kilodaltons. If you lay the DNA flat, it's 20 nanometers across, but again, it has that curvature, so it's probably more like 18 and change. And again, I think we're very optimistic about what it's going to look like because of the solubility and stability characteristics that we've seen under fixation, right? That being said, we've got to run the experiments. So,
we have one more question. How long would the dye last if the fixative was washed off?
we don't even know. I mean, I think we've stored ours for many, many months in a fridge, and as long as they're light protective, we don't see any degradation at all. And so it's a good question. You know, I think it'd be good to know over a period of honestly months, but it's incredibly stable. So we just haven't been able to test out as long as we'd like underneath either fixative or fixed wash leave-in PBS, you know, in light protection. I honestly would worry more that you would honestly evaporate the solution before these things would destabilize, because again, they're incredibly soluble and they're stabilizing that fluorescence. We just haven't tested out, you know, 3 months, 6 months, how long is that going to go for? Don't know yet.
Okay, another one, would serum release DNases degrade the probes?
Yeah, good question. So I'm going to break that down to one. So we do have some data on whole blood staining and didn't see any degradation there. So there's obviously some serum in that. I don't think we've tested serum alone. Is that right, Seddon?
Not separately.
Not separately, yeah. And then, but you know, the DNases in serum are pretty low concentration compared to what we use for staining. I will say that, I mean, if you're using something like DNase in a collagenase DNA solution, you've got a very, very high DNase concentration. The DNase will not affect the labels themselves, right? So it will not, because it can't recognize that actual phyton structure. What it will cleave, however, is the way that we're attaching that to antibodies, right? Which you could see that through, there's two different phases. This is sort of the Janus part of this platform. The bad part of that is, right, is you could, if you had very high DNase and you didn't wash it out, for instance, then you'd have cleavage of the dyes. The flip side of that is that you can use DNase and you can think about that in more of a sequential staining context.
Okay, I've got a very specific question sort of following that up. So it was following to the wood serum release DNases degrade the probe. If you were looking at netosis, for instance, would you need DNase-free serum?
Again, you know, I hesitate to speculate without any data on serum alone and the different DNase-free versus non-serum. So I think, you know, honestly, we'd have to test it, because I don't like, I think now I'm further out in terms of speculation than I want to be in terms of how that performed. Is that fair, Seddon?
Yeah, I agree. The next question is interesting. I would say that the question is, have these dyes been used in vivo like CD45 FITC to measure cells in the bloodstream? Some of the answer to that is that right now, even our access to mice, you can imagine with most facilities, it's kind of hard to get access to do that experiment. I think it'd be interesting. But
I think it would be interesting as well. I wish we could get there sooner than we can. But yeah, we're not. It'd be very difficult for us to do that right now, particularly because we don't have, you know, we certainly don't have a mouse house in our facility. I would say, you know, to that point, if that's something that's of interest, be happy to discuss further around what you'd like to do specifically, and if we could test that together, because again, our access is very limited to you know, particularly to animals.
All right.
Cool.
That's it. A lot of good questions.
Great. Yeah, please continue to feel free to use the Q&A function while I'm talking, Exeter and Seddon will interrupt me. But those are great questions. It's very interesting. In the serum, both the serum and the in vivo these are interesting things to think about. I think the serum one is an interesting one to test too, because I just don't remember off the top of my head what the DNase concentration in serum is as well, and whether or not we would see degradation there. So it's a totally very fair question. It's worth us looking into it, and the timing on that's quite good, because we're trying to do some of these experiments along those same lines now. Okay, so the third part of this is really a new to world capability, which is around controllable brightness, right? And what if you could take any color, and if you will have a volume knob on that? And so we've actually proven this out. So and I'll show you the first set of this data. And what we've done, we think about each one of these arms on the phyton as a cluster. Right, and again, I indicated that we can engineer down to sub-nanometer level the way these things work. right? But again, we also have 4 arms on which we can actually tune the brightness for these. For what it's worth, everything I've shown you, right, and everything that we've got in our commercial products actually has these 2 clusters. And the reason we did that is for, for the performance that you saw earlier, right, which is that we can get essentially at current dye level performance for what we've got now when we operate in the context of this 2 cluster. It's also how we develop dyes, right? We develop them in this 1 cluster format, and then generally speaking, we say, oh, they need to be brighter. Okay, we can go up to this. We've also proven out our ability to go up to 4 cluster. I'd say it's very much still in R&D, right? And so we're, I won't share data or discuss that too much, but I want to give you a sense of what we're looking at and how we're able to tune brightness. And so the first part of this is the good science experiment, which is, can you replicate the network that you have? And so you can see here, this structure is essentially symmetric along the diagonal axis. And so what we can do here is actually replicate that network. You can see, it's very consistent, very reproducible what we get in terms of that fluorophore network. And that replication is actually what enables us to tune brightness. And this is what's important about this. It's what enables us to tune brightness stoichiometrically, right? So it's not, ooh, it's 1.8 maybe. It's 2 times as bright when we look at it on a spec, right? And I'll show you the flow cytometry data in a moment. But I think this is important, right? Because it gives us the ability to really fine tune that. There's another important point that we don't think about that much, but when we attach dyes to fluorophores, generally speaking, we get a Poisson distribution of those dyes on the actual antibody. We try to work to really make that incredibly consistent in the context of conjugation, right? How do we get something more like 1 with a tight distribution, 1 phyton on 1 antibody? But the other part of this is what's in the dye. When we say, for instance, hey, that's going to be a 2-cluster Nova yellow dye, every single phyton, every single dye, if you will, fluorescent label in that tube is that 2-cluster label. There's no variation at all, right? And that's because of that assembly that I showed you earlier. So again, taking it back down to earth with this, now we can go twice as bright. What does that mean, right? And what it means is when you got to look at the flow, right? Because plenty of us have seen, I always say spectral plots are great. The spec plots are great, but they might as well be sci-fi. Show me the flow, right? And so what we're comparing here is a 1 cluster versus a 2-cluster Nova blue dye. We've got this fluorescence -1, right? We've got label only, so we've just doped in the 1 cluster and the 2-cluster labels. We've got low background there. And then what we want to do is compare those. And so what we see here, again, we've got the 1 cluster label in this dark blue and 2 in this pink, is we actually, we got twice as bright here. But I think what's important here is we've also increased the separation index, and we haven't changed the background at all, right? And I think that's a really important notion, is that we're not just tuning brightness higher and at the sake of less separation. We can actually tune the separation index. So what we can do when we think around dye design is actually say we want something to be this bright, and by that I mean the separation index of a population with low background. And that's what we're engineering for. So hopefully I've shown you these 3 capabilities. I was actually going to show you some brand new data as well that we've been working on in the context of a blocking solution that we've actually been working on as well, right? And so, but hopefully I've shown you this: bright and clean, consistent and stable, and controllable brightness. And so just to add some new data on it, because everyone loves the smell of new data, if you will, is I was going to show you some new data on a blocking solution that we worked on. And so for all the things that I've described and also the 40 color data I'll show you, there's actually only 1 change in workflow, right? Which is the same that you'd have to use for any of the PE conjugates or any of these APC conjugates, which is that we have a blocking solution for monocytes and macrophages, and you put on 5 microliters for 5 minutes before you're staining, and then you go right into your stain, right? No spin. And what does that mean, and how does that compare to other things? Like, what if you're working on monocytes and macrophages, or you have this background issue? Why else would that be important? So what we've done here, again, this is some brand new data. Setting just got to me at the end of last week, which is, we've got this gating here. We're just looking at singlets, and we're looking at all leukocytes here. And what we're comparing is no block, our block, and then the BioLegend TrueStain, which is this monocyte blocking solution, which takes care of this background that you see with these Cy and these Cy-based dyes. And so what you can see with no block, of course, you get this background, right? That's again from monocytes and macrophages binding up to these Cy-based dyes, actually using a particular receptor. And what we've done is design a blocking solution that's non-protein, it's not Fc receptor blocking, right? It blocks another receptor entirely, non-protein, non-antibody based, and it performs exactly the same essentially as BioLegend TrueStain, right? So it can block up the Cy-based binding of these dyes, same workflow as I described. we looked at that in that context. And of course, we looked at it in the context too of just looking at about monocytes and macrophages, right? So this is CD14 here, and you can see no block, right? You get this high background, but we're able to tamp that back down with this blocking solution. So just some new data I thought was worth sharing. Again, we don't have to store this. There are no other special buffers in using, you know, the Nova dyes, but similar to these Cy-based dyes, right? We do recommend that you use a blocking solution, particularly if you're looking at monocytes and macrophages. Okay, so now transitioning, you know, I think Rafe teed us up really well. I said we've got this spectral instrument. We're, you know, really excited around leveraging those capabilities, and so that's exactly what we'll talk about now. And so what we did is we started with this Cytec backbone panel, right? So they've shared this panel, 35 colors, 35 antibodies, 34 of them are commercially available. So we went out and got all of them and used that as our 34 color backbone. And what we did is actually drag, if you will, I always say drag and drop, but that's not exactly what we did. We dropped in our 6 labels, the 6 labels that we've been describing, and see if we could upgrade those panels and get it all the way to 40 colors, right? And for a number of different reasons. One, we want to look at dye-dye interactions, but 2, we just want to see, okay, well, what's possible when you have this kind of clean fluorescence that's not reliant upon signatures. There's another point in here, which is that we're not just using high antigen density markers. This also includes chemokine receptors. So we'll discuss that in a moment. And again, although this is probably the busiest plot I've ever shown on any slide, the important point is that we're hitting these detectors really, really cleanly. And so this is something that's quite important. So let's go in and talk about why that's important, right? And so what we did is we quantified the spillover spreading of just the 34 color backbone, right? That's what we're dealing with. And then Sen ran a really elegant experiment where she dropped in one of our labels at a time. So if you will, this new label is going to be the perpetrator and all the other dyes would be victims, right? Because if we're contributing spread into that, we're making the analysis harder across all other 34 colors, right? So what does that look like and what are the recommendations that come as a result of that? So again, drop in each one of these panel labels at a time, look at the spread, see if there's anything that's made worse. Maybe there's areas where there's no spread contributed, and let's figure out what that means in the context of panel design. And the way we did that, for what it's worth, was calculate a new spillover spreading matrix for the 34 plus 1 and compare that to the backbone. So a very simple experiment. So the first thing we see is NovaBlue 530, which is very similar to Brilliant Blue and FITCI, contributes a lot of spread, right, into those channels, particularly in the BB 515. But that's exactly what we'd expect, right? Brilliant Blue 515 and FITCI are very, very difficult to separate, right? We've had huge challenges in separating those. So what we would actually recommend is actually using NovaBlue 530 and BB 515 or NovaBlue 530 and FITCI, because they're easier to separate, right? And it lowers the complexity of the actual panel, but it's exactly what you'd expect. The other important part is what you're not seeing anywhere else in the slide, which is that you're not seeing any spread contribution anywhere else from NovaBlue 530. You're seeing nothing with 610 and nothing with 660. So in other words, you can upgrade the number of questions you're asking in a panel and contribute no spread anywhere else. And I think that's a really powerful thing to consider. And again, we'll bring this back around because we are looking at some lower antigen density markers here. Let's transition over and look at the Nova yellow dyes. And actually in this case, it's exactly what you would anticipate. Again, note that the y-axis, we're not contributing huge amounts of spread here. But what we see is that each yellow dye contributes some spread to the dye that it's most similar to, right? With the B570 and looking at, you know, contributing some spread in a PE, Nova Yellow 610 and a PE Dazzle and 660 and PE Cy5, right? But nowhere else are you contributing spread, right? And I think that's what's important about this experiment. It's what you're not seeing. And the other part of this too, for what it's worth, is that, you know, these are intended to replace these dyes on a conventional cytometer. So that's the importance of this. But it's that, you know, and the other part is when you're thinking around your spectral panels, you know, whatever your backbone panel is, essentially what I've handed you is the ability to answer 6 more questions, right? Because of this low spread contribution. So what's high resolution biology in this context, right? We've added this 6 colors with essentially marginal impact on spread, with the exception of a very difficult combination already, right? And we talked to a number of different people running spectral panels, and they echo, you know, what we've seen on this BB 515 FITCI. And really, you know, the goal there is, can we get towards easier experimental design? Can we get higher resolution of biological populations? And so that's also what we attempted to show by both sharing the data and all of the analysis. We also created this subway plot for expressing how biological populations are related to one another, how we gate, because I've just honestly been frustrated for years around how we communicate our gating hierarchies, and was trying to come up with something that was a little bit better. And so this has had a life of its own, and we're free to share this PowerPoint template if you want to you want to use this for your own biology, happy to do that. You know, whether you're interested in our NovaFluors or not, I think it's a hopefully useful way of communicating. But I think the important part about this, when we think about all the 32 populations that are at least manually gated here, is that what we're able to do is layer on some more interesting biology by looking at these chemokine receptors, by looking at these low antigen density markers, using these high resolving fluorophores, and do that across all 32 populations. And that's exactly what we see here. And so I think that's what becomes very interesting, is your ability to leverage these clean fluorophores to do some more interesting biology. To that point, you can actually take all these manually gated populations and project them. And what I've shown here in gray is actually the populations that just aren't gated, that are at the lymphocyte level, right? So there's a lot more biology, interesting biology, you know, to discover here. And I think that's what's really interesting. And that's what also drove us to, you know, not just put up a white paper, which is great, but we also shared the unmixed data, the raw data, as well as all the data analysis and how we did the experiment. And so to us, that's what's important. And because again, regardless of honestly if you're going to use NovaFluors or not, we think this is a really good resource for panel design. How are these dyes interacting? And hopefully it addresses some other questions, right? If you are thinking around these higher end panels, is, well, how does this work with the brilliant dyes? How does this work with brilliant buffers? You know, this is part of that answer to that question, right? Which is that we did push this all the way up into this 40 color range. How are we doing any of this, right? Like what does this look like? You know, how do you work with us? So what we've done is we've put out 2 different CD4 test kits, one on human SK3, the other one on mouse RM45, right? So that enables you to look at these, again, across a number of different flow cytometers, look at SSM, look at how you might include these into your panel. We've also wrapped these in a 6x100 microgram conjugation kit. So you can take, you know, 6 of our dyes and conjugate up to 100 micrograms of those individual antibodies. We've also taken on some custom conjugation projects, right? If this is something that's of interest, happy to discuss, right? We take, you know, 1 milligram or up and conjugate those. Maybe I'll get ahead on a question, which is, are you looking ahead towards providing your own conjugated antibodies? And the answer is absolutely. It can't happen soon enough. I'm hopeful that we're going to have a lot more to say on that very soon, but it can't be soon enough for me. But regardless, you know, I think we've got a really nice answer for the ability to expand capabilities right now. And so, you know, the other part was we've actually broken out our conjugation kits into individual colors as well. And this is what's enabled us to work with a number of different people as well, because they'll say something like, I want to drop, you know, can we drop these in? Can we drop 2 of these colors in and then, you know, sort of upgrade a panel and add more capability? It's been very useful for Seth and I as we have those conversations. And so we're enabling that as well. And so hopefully what I've been able to show you is, you know, we've got bright and clean labels, consistent and stable. They're tunable, plug and play, and you're able to discover more. Happy to discuss more around the conjugation kits, any other questions you have around the fluorophores, but I want to respect some time and save a couple minutes for more discussion. So yeah, with that, we'll just open it up.
Okay. So you already answered the question about providing labeled antibodies. So there's an additional question. Any scope of developing dyes that are excited by the UV or the red laser?
Yes. Stay tuned. I didn't intend for that to be a pun. So the answer is absolutely. And so I think you've immediately seen how quickly we can do that. It's deterministic enough in terms of the new labels that we're developing along the pace that we're doing it, that I think it's June 23 and 24. We actually will be launching a set of fluorophores that'll answer your question directly. And, you know, which laser lines and what other labels are you doing? And we'll show every single one of them, right? And so I would say it's not even stay tuned. It's like there's a date. And so you can go sign up and say that's something that's very, very much of interest to me.
Nothing else right now.
Okay. Well, we're happy to hang out and answer questions and talk about fluorescence and panel design. But maybe before that, thank you so much for the invitation, Rafe. It was really, really fun, and hopefully we get some more discussion going here as well.
Yeah, got. So one question: what is the Nova block?
Yep. So non-protein, non-antibody based. It blocks. So we went in and characterized. There's a receptor on monocytes and macrophages, which is CD64, right, which binds on to these Cy-based dyes. And so, and that's been shown in the literature. And so what we created is that blocking solution that just blocks that up. And so, again, non-protein, non-antibody based blocking solution that'll essentially bind onto that receptor and prevent what you saw, prevent the side-based dyes, prevent any NovaFluors from binding onto those receptors. So it's not just scavenger receptors, for instance, on monocytes and MACs. It is a specific receptor that's being blocked.
Okay. 2 questions about the conjugation. One of them, what if the conjugation fails in the user's hands?
Okay, good question. We haven't seen that to date, and the chemistry we're relying upon, at least for the first, for these first set of kits, we're relying upon a partner for that, and they've tested that chemistry in thousands of kits. I'd say, it'd be I think in that case, Seth and I have been very focused in on not just, does the kit work, but then enabling success on the other end of that, which is getting it into the panel. I think what we'd want to do is get in and quite literally troubleshoot with you and find out why that is. I'd say, we've been aiming for a very low turnaround time on kits, right? So, really one evening and then you're back in the, one evening incubation and then you're back in the next afternoon and can use it. And it has 5 pipetting steps in the current formulation. So, I think if there's an issue along that, with pretty well understood chemistry, I think we've got to get in and dig and understand what antibody or what protein are you trying to conjugate to and figure that out. We haven't run into that to date, and we've been playing with this conjugation and using this conjugation for quite a while. So that'd be my answer, because we just haven't run into it yet.
A follow-up, does the conjugation use non-amine based chemistry? I need to label a fusion protein and this will activate my functional site.
I want to understand that question a little bit better. Yeah.
Yeah. So I think I can maybe answer part of it. It does, at this point, the conjugation kit does use an amine-based chemistry. We could definitely work with you. We actually have a very smart conjugation chemist. There are obviously other approaches to label things. So that's maybe something we could talk about if that's something you're specifically interested in. So,
yeah.
Can you conjugate the dye to any other functional groups such as avidin or any particles such as iron?
So we've never done an avidin. We haven't done one yet. As long as there's free lysines on there, right? Then we'd be fine on the avidin side. On an iron particle, depending on how the iron particle is activated, right? And what chemical groups are hanging off the iron. But have we done it yet? No. I mean, transparently, if I don't have data, I'm not going to tell you that we've done it yet.
All right. Any plans for sort of a follow-up to that? Any plans for in vivo tracking applications near IR option?
Yeah, it's very interesting. I mean, it's an interesting question, because when we showed the platform here, particularly the ability to solubilize dyes and move the brightness up, folks who had IR-based dyes became very interested in this. This was a while ago now. And so I would say that's an area of interest for us. I think there's a lot more work for us to prove out that the platform works in vivo sort of a priori. And then I think there's a lot of interesting, honestly, component dyes that are out there that have been underutilized, both for in vivo applications and in other, particularly in flow and other applications, because they just haven't, the dye performance itself has been a limiting factor there in the IR. So I'd say, I get very interested in the IR because of, I think, to your point, with its in vivo imaging applications, but I think we need to do the first principles experiments about what possibly can we do in vivo, right, and at what level, and then go back in and say, okay, now that we know that, let's see what's out there, right? But we had some very interesting conversations early around, well, what could that mean?
No other questions right now, but I want to further reiterate, I really appreciate these great questions, because Mike knows I keep a list of questions, of things we want to test out and try. So it's always great to get questions that I haven't necessarily thought of as we're going forward.
Most of these are ones we haven't been asked before, which is fantastic. I think there's a lot of interesting application questions that are coming out that are not just, if you will, not just flow, but flow and or fluorescence and that's a very meaty area for us to dig in on and understand. Yeah.
That means we have a collaboration. This
There you go.
application.
Yeah. No, it's very interesting, right? I mean, it's very cool questions, particularly the in vivo stuff is very intriguing and just not something we know, right? And again, it's
Yeah,
one of those things.
I mean, that's really, thanks very much for the great talk and for the nice presentation. So I really enjoyed, and also to see the different type of dyes and the different technology. And actually, we got the spectral flow cytometer from Cytac, and I'm really keen on test on these different panels, and also very keen on testing the NovaFluors as well. And I'm sure there's a lot of colleagues in the immunology field, also in different fields, to how they will be able to use it as well. But there's a lot in Exeter around also biological applications. That's why this bacterial or iron oxide particles, and I'm pretty much interested in this type of applications as well. So it's a great opportunity, and I hope you guys have enjoyed as I did. And many thanks, Mike and Sedan, for organizing and for giving these nice talks. And yeah, hope to see you all in the future again.
Great.
Yeah. So any follow up, you can contact also Mike directly, but go also via our Cytomics Center. Right. Thank
Great.
you.
Thank you.
Mike.
Oh, yeah, and one last, yeah, you got it. The email address, info@phytonics.com, if you have any questions for us.
Sure.